lymphocytes are a crucial component of the adaptive immune system, primarily responsible for humoral immunity. They produce antibodies, which are essential for neutralizing pathogens and toxins. Here's a breakdown of their structure, types, mechanism, and function. B cell structure. The most distinctive feature of a B cell is the presence of thousands of B cell receptors, BCRs, on its surface. Each BCR is a membrane-bound antibody molecule with a unique antigen binding site. A typical B cell receptor is composed of 4 polypeptide chains, 2 identical heavy chains and 2 identical light chains. These chains are held together by disulfide bonds. Antigen binding regions, formed by the tips of the heavy and light chains. These regions are highly variable and allow each B cell to recognize a specific antigen. Transmembrane proteins. The BCR is associated with a complex of transmembrane proteins, Ig-alpha and Ig-beta, that are crucial for transmitting signals into the cell when an antigen binds. These proteins have short cytoplasmic tails that link to intracellular signaling pathways. The heavy chains determine the isotype of the antibody, e.g., IgM, IgD, IgG, IgA, IgE, which influences the antibody's effector functions. Immature B cells initially express IgM and then co-express IgD on their surface as they mature. Types of B cells. B cells undergo various developmental stages and differentiate into distinct functional types. Transitional B cells. These are immature B cells that have recently left the bone marrow and are migrating to secondary lymphoid tissues. They are a link between immature and mature B cells and undergo further selection processes. Naive B cells. These are mature B cells that have not yet encountered their specific antigen. They circulate through lymphoid organs, waiting to be activated. Plasma B cells, or plasma cells. Upon activation, naive B cells differentiate into plasma cells. These are large, antibody-producing factories with a highly developed endoplasmic reticulum. Their primary function is to secrete large quantities of soluble, antigen-specific antibodies. Plasma cells are generally short-lived but provide a rapid initial response to infection. Memory B cells. After an initial encounter with an antigen, some activated B cells differentiate into long-lived memory B cells. These cells remain in the body, often for years, and can quickly reactivate and differentiate into plasma cells if the same antigen is encountered again. This memory is the basis of long-term immunity and vaccine effectiveness. Follicular, FO, B cells. The main subset of B cells found circulating through the blood and in lymphoid follicles. They are responsible for generating the majority of high-affinity antibodies during infection, often requiring T-cell help. Marginal zone, MZ, B cells. Located in the marginal zone of the spleen, these B cells serve as a first line of defense against blood-borne pathogens. They can respond to antigens without direct T-cell help, T-independent activation, and produce rapid, but often lower affinity, antibody responses. B1 cells. A distinct population of B cells found in specific tissues like the peritoneal and pleural cavities. They have different developmental origins and often produce natural antibodies, IgM, that provide immediate protection against common microbial components, even without prior exposure.